Hello everyone, welcome to Aakash. This is Lavina saying your botany faculty, and here to continue with the second slot. You are already done with this first slot of this test paper, that is AIATS Plus Phase 2 Mock Test 9, which was conducted on 16th of March 2025. So you are done with the questions from the first slot, which contains several chapters. Now I will be taking up for the second slot, which will be considering these very chapters, that is biological classification, anatomy of flowering plants, plant growth and development, molecular basis of inheritance, and biodiversity and conservation. So the questions I am going to discuss will be the ones in which you make frequent mistakes, and they will be from these chapters. Alright, your NEET exam is overhead. I hope you guys are preparing well, you are all buckled up, you are polishing your last minute strategies, your last minute concepts, right? And it is very important at this very moment that you again go through your NCERTs for the final touch. Alright, so very good morning Kumaran, and today I will be taking up with the next question that will be continued from the first slot, then now the second slot. So we have the question, that is question number 92. It is from biological classification, which says diatoms differ from dinoflagellates as the former. So in the question we have been asked about the former, that means we are considering diatoms. We have to see that which characteristic feature is true for diatoms, and it is a differentiating feature also with dinoflagellates. Okay, so but before that we need to understand that from where these two come from, right? To which particular category do they belong? Alright, very good morning Xenia. Now we will understand it by again classifying the kingdom Protista. So the main kingdom that we are considering, that is the kingdom Protista, and this Protista kingdom is having the organisms or the members that have a very precise and particular feature that is applicable for all its members, that is they are unicellular eukaryotes. Right, I have discussed it earlier also with you, and this particular feature, it should be very much clear with you, because this unicellular eukaryotic feature helps you solve many problems, many questions, because usually they are asking you questions which of the following is a eukaryote, or maybe the examples will be given, right? Or unicellular eukaryote, you have to pick the answer from the Protista kingdom, right? So I hope this is clear. Now kingdom Protista, it is divided into three categories, right? The first one is of photosynthetic protists. Right, that means these will be performing photosynthesis, they will be preparing their own food in the presence of light, so they are the photosynthetic protists. If we consider the second category, it belongs to that of consumers, right? They cannot prepare their own food, and also they are basically decomposers, okay? So they are saprophytic, they are the decomposers, right? They feed on the dead and decayed matter. And the third category is of the protozoans, right? Now protozoans are also the consumers only, but they are not the decomposers, okay? That is why we are putting them in the third separate category, all right? Now the question is about dinoflagellates and diatoms. So they are falling in this very category, right? So if we want to write the examples, we can consider photosynthetic protists. We have diatoms, we have dinoflagellates, right? Euglenoids are also falling in this very category, although they show mixotrophic mode of nutrition, but we can consider them as photosynthetic also, because in presence of light they will be performing photosynthesis. Then consumers, which are acting as the decomposers, we have the slime molds. And the last one, we have so many different categories of protozoans, right? They are ciliated, flagellated, they can be amoeboid, right, as well as sporozoans like Plasmodium, right? So this is the categorization of kingdom Protista. We have to focus on these two, that is the diatoms and dinoflagellates. Now we have to see that what are the characteristic features, okay? So I will be discussing some common characteristic features, okay? So I'll be discussing them over here. So first of all, diatoms and dinoflagellates. Right? What are the common features between them? First of all, because they are belonging to the kingdom Protista, they are unicellular. Both of them are unicellular. Alright? Secondly, we see they have the presence of cellulose in the cell wall. Right? So we can say cellulosic cell wall. This is a common characteristic. Then the third characteristic is both of them are photosynthetic, right? So they are having pigments, right? So the pigments that are present are chlorophyll A, chlorophyll B, they have xanthophylls, right? As well as carotenoids, right? So these are the pigments that are found in both of them, right? This is again a common characteristic feature. Then next common characteristic feature is if we consider asexual reproduction, okay? So asexual reproduction in both the cases takes place by binary fission. Okay, so these are some of the common characteristics between the dinoflagellates and diatoms, right? Now moving on to the options. All right, hello Sagnik, कैसे हैं आप? अच्छा ओके सत्यनारायण, मैं बीच बीच में हिंदी बोलूँगी ताकि आपको समझ में आए. All right, very good morning to all of you. Alright, so हमने अभी ये देखा diatoms and dinoflagellates, what are the common characteristics. Now switching to the options, first have cellulosic cell wall. We have to see that why they differ from each other, right? Why they differ from each other. So cellulosic cell wall दोनों में होता है, so this is not true. Then lack chlorophyll A. अभी मैंने आपको बताया that chlorophyll A is present in both diatoms as well as dinoflagellates. So this is also not true because this is a common character for both of them. Then float passively in water currents. That is absolutely true because when we are speaking of diatoms, they do not have flagella, right? While we are considering diatoms, they are non-motile. Right? They are non-motile, they do not have flagella. So they float passively in water, and that is why we can also call them as planktons. Right? Very good Sagnik, they float passively in water. Right? So this is the correct option because this is not true for dinoflagellates because as the name indicates, dinoflagellates, they have the presence of two flagella, right? One, they are also called as the whirling whips. If you remember, they have two flagella, one is longitudinal, other is transverse, okay? So they do not float passively in water. They are showing locomotion with the help of flagella, right? Dinoflagellates, but diatoms float passively in water. That is why these two differ from each other, right? And the fourth option is inhabits aquatic habitat. Obviously, both are found in aquatic habitat, okay? They can be found in fresh water, they can be found in marine environment also. So both of them are aquatic. So it is also not a differentiating feature. So the correct answer for the given question is our third option. I hope this much is clear with you, right? Whenever such questions are asked, always focus first on the common characteristics, okay? Then you will automatically come to know that which one falls for the different, okay? Now, the next question that we move on to is question number 97. all of the following structures are absent in the lamina of dicot leaves except, okay. So the question is from anatomy of flowering plants. When it comes to anatomy, a lot of you make a lot of mistakes, okay. I know it's a bit tricky chapter. There are so many concepts that you have to keep in mind. What is conjoined vascular bundle, what is closed vascular bundle, right? What is the abaxial surface, what is the adaxial surface? These are some of the concepts in which you make frequent mistakes, okay. So we will go through the structure of a dicot leaf, okay. What is the anatomy? A brief explanation of that so that you understand it in a better way, all right. A quick revision of the concept, because in this very series we are not only discussing the answers for the questions, we are also discussing the related concepts, right. So let us understand the concept. Here we have the structure of a dicot leaf, right. You can clearly see there is the presence of the, first of all, we can simply divide the structure in three parts. First of all, epidermis, second is the mesophyll tissue, and third one is vascular bundles. Okay, one by one I will discuss it with you. All right. So first of all, we have epidermis. Now, see, while we are considering root or the stem, we have only single layer of epidermis, that is the outer. But in case of leaf, we have two surfaces and hence two epidermis. Okay, so while we are considering, epidermis, so first of all, it is single layered. Okay, single layered. It is parenchymatous. Okay. Second characteristic feature is it is parenchymatous. And the third very important feature is they have the presence of a layer of cuticle. Okay, you can see in this representation also, this is from your NCERT. Here we have the pink colored epidermis, and above that there is the thin layer that is green in color of cuticle. Okay. Yes, very good, Sagnik. There is the presence of the upper and lower epidermis, that is the adaxial and abaxial epidermis. Okay, so single layered epidermis is single layered, it is parenchymatous, as well as they have the presence of cells that are cuticularized. Okay, because of the presence of cuticle, we are calling them as cuticularized. All right. Now we are dividing this layer into two regions, right? That is the upper epidermis and the lower epidermis. So the terms that we use for upper and lower, because upper and lower is general term, okay, we have to give them a specific term. So the terminology that we take up for upper, we use the word adaxial. Please keep that in mind, okay. We make frequent error in this, which is adaxial, which is abaxial. If you are seeing a leaf, the shiny surface that is present on the front, that is on the upper part, okay, that is the adaxial epidermis, the upper epidermis. And अगर आप उसके back की तरफ देखोगे, right, that means the back of the leaf is a bit light colored. Okay, we can see the venation in that. It is a bit rough also. Okay, so at the back of the leaf, it is abaxial surface. Okay, that is the lower side. Okay, so the lower epidermis. So it is the adaxial one, and this is the abaxial one. All right. Also, on the abaxial surface, we observe numerous stomata, right? Because stomata are, they are meant to exchange gases, right? There is the presence of chloroplast in the guard cells. They are meant to perform so many functions, right? For transpiration also they are used. Now, if the stomata are exposed, right? Suppose this is the leaf, sunlight is falling like that, okay, so it will harm the stomatal apertures, okay, or what will happen, a lot of transpiration will take place, the heat is more over there, right? That is why the plants have the presence of stomata. More stomata are present on the lower surface of the leaf, okay, so that they are a bit protected. All right, so that is the abaxial surface. Okay, numerous stomata are found on the abaxial surface. This question is also asked. All right, now this is about epidermis. Now moving on to the second part, that is the mesophyll tissue. Okay, so mesophyll, while we are speaking of mesophyll, the term is divided such that meso means middle, phyll means leaf. That means the tissue that is present in the middle of two epidermis. This is the upper epidermis, this is the lower epidermis, and the tissue in the middle is the mesophyll tissue, middle leaf, right? So mesophyll tissue is basically again divided into, it is parenchymatous tissue. Okay, so there is the presence of two types of parenchyma. A very good morning to you, Pankaj. So here we have the palisade parenchyma, and the other one is spongy parenchyma. इस डायग्राम में आप देख सकते हैं, right? The palisade parenchyma किस साइड प्रेजेंट है? ऊपर की तरफ प्रेजेंट है. ओके, सो ऊपर वाली साइड प्रेजेंट होने की वजह से इट इज adaxial in origin. Okay, so palisade parenchyma is adaxial. That means it is present towards upper epidermis. Secondly, the cells. Now you can see the structure of these cells. The cells are elongated, right? Yes, Hema Malini, spongy and palisade parenchyma. So palisade parenchyma is present towards the upper surface, that is the adaxial surface. Secondly, the cells are elongated, right? They are present parallel to each other. You can see they are present like this. They are elongated and they are present parallel to each other. Okay, they are parallel, right? And they have a very little intercellular space. Okay, so they have little intercellular space. That means they are very closely packed. Okay, they are very closely packed. And the third characteristic feature that we can consider is they are rich in stomata. Okay, they are rich in stomata. Sorry, they are rich in chloroplast. I'm so sorry, they are rich in chloroplast, because this is the tissue that is going to perform the photosynthesis. So there has to be the presence of so many chloroplast over there. Okay, yes. Spongy and palisade parenchyma, very good Momita. This is a characteristic feature particularly of the dicot leaf. In monocot leaf, there is no such distinction. Okay, so the distinction is seen in this case only. That is why I told you in the very beginning that we are discussing the structure of a dicot leaf. Okay, so this is the diagram from your NCERT. Now, if we compare it to the spongy parenchyma, we see in case of spongy parenchyma that the first characteristic feature is differentiating because it is abaxial in origin, right? It is present towards lower epidermis. So it is, first of all, it is abaxial, right? Secondly, if we compare it with this one, the cells are not elongated. The cells are oval or spherical. Right, oval or spherical cells. Also, in this case, these are parallel, right? They are not parallel, they are just scattered. Okay, and third, they are not closely packed, they are very loosely packed. Okay, so the cells are loosely packed. And why they are called as, why this particular parenchyma is called as spongy parenchyma? You have seen a sponge, in that there is a presence of air spaces, a lot of air spaces in the middle, right? So we have the presence of air spaces. You can see in the representation also, right? These cavities that we can see, they are the air cavities. Okay, so the presence of air cavities between the cells, right? So this is the difference between the palisade and the spongy parenchyma. This is the mesophyll tissue, right? Now we have to see the other one, that is the vascular bundles. We have discussed epidermis, we have discussed mesophyll. The third one that is there are the vascular bundles. Now in case of leaves, the vascular bundles, they are closed because leaves do not show secondary growth and cambium is absent, and if the cambium is absent, we call it as a closed vascular bundle, right? Okay, now we have the closed vascular bundles. Please do not spam the comment box, all right? Yes, so we have closed vascular bundles, right? And we see that in case of dicot leaf, please pay attention, I am discussing dicot leaf with you. So, the vascular bundles vary in size. Vascular bundles vary in size. Why? Because in case of dicot leaf, we see reticulate venation, okay? Due to reticulate venation. Now, what is that? See, in case of a dicot leaf, what do we observe? There is a midrib, right? There is a midrib. I suggest you to imagine the leaf of peepal, okay? आपने कभी देखा है पीपल के पत्ते जब झड़ जाते हैं, जब नीचे वो ड्राई हो जाते हैं, आप लीफ को उठाओ, प्रॉपर उसका skeleton नजर आता है आपको, right? A huge network you can see, very beautiful, right? Now imagine that. So there is one midrib that is a bit thick as compared to the others. Now there will be the presence of veins, right? There will be the presence of veins, and further there will be the presence of veinlets, right? So there will be the presence of veinlets also arising like this and forming the whole network, right? So this network, as the veins are thicker as compared to the veinlets, so if we see, if I dissect this part and see the midrib portion, the vascular bundles will be a bit broader, okay? They will be larger in size, okay? They will have more diameter. But if I compare it to the vascular bundles in the veinlets are very thin, so the particular vascular bundles will also be very smaller in size, right? So this is the difference. The size of the vascular bundle varies because of the reticulate venation, right? Clear with this point? Alright, अगर कोई भी doubt है, please ask me in the comment box. Alright, now the third characteristic feature that we see is these vascular bundles are surrounded by bundle sheath. They are surrounded by bundle sheath. Right? So these characteristic features should be very much clear with you. This is a brief discussion of the anatomy of the dicot leaf. Now coming back to the question. The question is asking which of the following is absent in case of dicot leaf. So can you answer from the given options whether it is endodermis, bundle sheath, pericycle or medullary rays? Which of the following is absent? Okay, we have to tell except. Okay, pay attention on except also. All of them are absent in dicot leaf except, that means which one is present, right? So you have to tell the correct answer. Sagnik, can you tell me in the comment box what should be the correct answer? Which parts we have discussed out of these? The last one that you should remember, obviously, that is option 2. All right. So bundle sheath is present in case of dicot leaf. There is no endodermis, no pericycle or medullary rays. Alright, so this should be clear with you. I will be discussing the tissue system with you in the question ahead. That will be very much clear that what do we understand by medullary rays. Alright, bundle sheath are found in case of leaves only, right? Not in case of stem, not in case of roots, only in case of leaves. But endodermis, pericycle and medullary rays, they are found in case of stem or roots. Alright, moving on to the next question. Question number 98. This is what I was about to tell you, that ground tissue system. Alright, Karthik Narayan is saying 1, which is not true. Please pay attention on the previous concept, okay, that I just discussed with you. Very good, Anshu Raj. The correct answer was 2. Now, question number 98 is about ground tissue system. Okay, before coming to these options, we need to understand tissue system in brief again. Okay, because again I am telling you, this is not the discussion of the questions only, it is also the discussion of the related concepts. Okay, so I will be discussing the tissue system with the help of dicot stem. Okay, so tissue system and here I am taking up dicot stem because it comprises of all the parts. Okay, so it will be very much elaborate and clear with you. So if we consider dicot stem, now tissue system is divided into three main categories. Okay, so the first category that we have is of epidermal tissue system, right? So we have epidermal tissue system. Second one is the ground tissue system on which the question has been asked, and the third one is the vascular tissue system, right? This we study in the lower classes also, right? So first of all, we have the epidermal tissue system. If we are taking up dicot stem, so what will be the parts of the epidermal tissue system? First of all, we have epidermis, obviously. Yes, epidermis. Had I been speaking about root, then this particular layer will be called as epiblemma, right? But I am telling you it is about stem, so we are using the term epidermis. Alright, now second one, yes Sagnik, that is absolutely true. You are already clear with the concept, just give it a quick revision. Alright, so epidermis, secondly we have the presence of stomata as well as trichomes. Okay, because it is stem, that is why trichomes. Had it been root, there will be the root hair. Okay, we can also call them as the epidermal appendages. Okay, so this is the epidermal tissue system. Now ground tissue system will comprise of, first of all, see, epidermis के बाद next layer कौन सी आएगी? In case of dicot stem, it will be hypodermis. Second will be the general cortex, and the third one will be the endodermis. Now all three form the region of cortex, right? So this is the cortex region. Then the next part that we take up is the pericycle, right? So we have pericycle, then the presence of medullary rays, इनको हम pith rays भी बोल सकते हैं, medullary rays, and the last one is the pith. Okay, so all these regions, they are together forming the ground tissue system. Alright, last one is the vascular tissue system, and vascular tissue system, obviously there will be the presence of vascular bundles, that will be xylem, phloem, and the last one, that is cambium, that will help in secondary growth. Okay, so this is the distinction. Now the question, back to the question is asking the ground tissue system. First option says is composed of simple permanent tissues. Absolutely true, right? There will be the presence of parenchyma, collenchyma, sclerenchyma, but no complex tissue like xylem or phloem. So this is absolutely true for ground tissue system. We will also go through the rest of the options. Second option is composed of complex permanent tissue. No, there is no xylem or phloem in case of ground tissue system, that is in case of vascular tissue system. Third option is includes all the tissues except epidermis, right? Yes, epidermal tissue is not considered under the ground tissue, but it is also not considering the xylem or phloem, right? So vascular bundle, except epidermis, there is also vascular bundles, right? So that is also not the part of ground tissue system. So this is not true. Then includes general cortex, yes. Endodermis, yes. Xylem, no. Cambium, no. So the correct answer for the given question, as Sagnik told in the comment box, that is option number 1. Alright. So this is how you're gonna rule out the options by keeping the concepts clear in your head. Alright, moving on to the next question that we have. Question number 109. The plant growth regulator that can be used to produce parthenocarpic fruits in tomato is, alright. So it is from plant growth and development. When it comes to the questions from PGD, plant growth and development, mostly the questions are asked from the section that is plant growth regulators, right, the hormones. Now there are so many hormones, there are so many roles that are being performed by them, right, their composition, their origin, their experimental discovery and everything, right. Off topic thing, okay. If I get selected in this year NEET examination, I would then like to meet with you along with other teachers, as I have only studied from you teachers through online mode. Wow, just wow, this is a very pretty, very beautiful comment over here, and we can maybe arrange a small get together with all of the students from the online mode, right. There is this ceremony that is conducted in which you can join maybe, right. So yeah, we will try our best for the online audience that we have to have a meet, right, with all the teachers, alright. Thank you so much, Sagnik. And I really hope and pray that you get selected in NEET in your medical college, right. Alright, yes, Sagnik, the correct answer. Anshu Raj ने भी सही आंसर दिया, मोमिता ने और सत्यनारायण सभी ने सही आंसर दिया है. That is indole 3-acetic acid, and that is what? It is an auxin. Right, it is an auxin. Okay, the rest of the roles that is responsible for bolting, it is which hormone? Can you tell me in the comment box that is responsible for bolting? We have to go through the rest of the options also, na? So the correct answer for this is gibberellic acid. Then a gaseous compound. The gaseous compound, only gaseous hormone is ethylene. Okay, so this is ethylene, and it is not responsible for producing parthenocarpic fruits in tomato, right. And the other one is released by ethaphone, that is again ethylene. Okay, ethaphone is a compound from which ethylene is released. Okay, so again that is not true. Okay, now auxin, here we have auxin, that is the correct answer. We have to discuss the related concept also, right. So what are the other functions that are performed by auxin? Now auxin is the hormone, right. Auxin is present in both forms, that is the natural one as well as synthetic one. Right? If we are considering natural, we have indole 3-acetic acid, and we have indole butyric acid. Then if we are considering synthetic auxin, we have 2,4-D, 2,4-dichlorophenoxyacetic acid, as well as NAA, naphthalene acetic acid, right. Now, these are the categories in which the auxins have been divided. Now, speaking about the roles that are performed by auxin in a plant. Apart from inducing parthenocarpy in the seedless fruits like tomato, we have other roles also. It is very important. Yes, 2,4-D and ethylene. Okay, now we have, first of all, if we are talking about rest of the functions that are performed by auxin, it promotes flowering in case of pineapple, okay, and lychee also. Okay, this is the first function. Okay, one more thing that I have to tell you is the functions that have been given in your NCERT with the respective hormones. Please do read them carefully, please memorize them, okay, because the questions are often asked from those roles only. Okay, so next is this parthenocarpy in tomato, that is also given in your NCERT. Okay, so promotes flowering in pineapple, alright, and it shows synergistic effect. Synergistic effect means if one hormone is performing some function, the other hormone is also performing the same function. Okay, that means they are synergizing, they are performing the role together. So that is the synergistic effect. And the opposite is antagonistic effect. That is one hormone is performing certain role, the other one is stopping that. Okay, so that is the antagonistic role. But here the synergistic action or synergistic role in cell division, synergistic role in cell division, और किस हार्मोन के साथ ये synergistic effect show करता है? With cytokinin. Okay. Next function that is performed is rooting. Okay. Suppose we have certain stem cuttings. We want to vegetatively propagate, let's say, rose. Okay, we have stem cuttings from rose. Now what we do, we take the stem cutting, right, and we put them in the soil. Okay, we simply plant them in the soil. So what we should do is we should dip them in auxins. Now what are the sources of auxin? For example, we have aloe vera gel. Okay, so you can take the leaf of aloe vera, simply rub that particular gel on one end of the stem, and that rubbed part should be put in the soil. Okay, that will promote rooting. Why so? Because aloe vera gel is rich in auxins, okay, as well as cytokinins. Okay, so that will help in promoting rooting process. Okay, so auxin is performing this very role. Other role that is performed is apical dominance. Very, very important. Okay, this question is asked. The hormone is important. Okay, what is apical dominance? This particular hormone auxin, it promotes the activity of apical bud. Okay, so the plant keeps on elongating. It is simply growing in length, alright. So the lateral growth is hindered because apical growth is taking place, the lateral buds are suppressed, and that is why the plant is simply elongating. Okay, and sometimes we do not want that. For example, if we want to make hedge, आपने देखा है जो gardens होते हैं, जो parks होते हैं, बहुत broad hedge बनी होती है grasses की या फिर बहुत सारे plants की. Okay, so that hedge formation, or in the tea plantations we can see. Have you seen the tea gardens? In those tea gardens, it is very important to make them dense. Okay, the hedges are dense. Okay, and that is done by cutting them from the top, because this hormone is found in the tips of the roots and stems, and if we cut the stems, the tips of those particular parts, what will happen? The growth will be, the apical growth will be then suppressed. Okay, because apical dominance is stopped now, right, and the function of cytokinins will take place and the lateral growth will be promoted. Okay, yes. It is present in the tip of coleoptile. Very good, Momita. Now, apical dominance is performed by this, and it is also acting as weedicide. Because auxins, basically, if we take the example of 2,4-D, right, it mainly hinders the growth of dicot plants. Right, and if the dicot plants are hindered from growing, we want to make a lawn. This is given in your NCERT, weed-free lawns. Okay, so the dicot weeds, they are hindered, right? The growth of them is hindered by using auxins, right? And the lawns are made up of grasses. Grasses are monocots, so their growth is not hindered. Okay, they are simply growing, they are simply flourishing. But we do not want the dicot weeds in between, so we simply apply auxin and prepare the weed-free lawns. Okay, clear with this? All right. If you have ever been to a nursery or ever bought a plant which is very, usually dense, for example, a Christmas tree, okay, we simply buy that, and the person that is there who is selling that, the gardener, he tells us, keep on cutting the tip of this particular plant, तो ये घना हो जाएगा, तो ये broad dense हो जाएगा. Why is he telling you to do so? Because the auxins will be removed, right, from the tips, and that is why the hedge formation will take place. Okay, clear with this? Now moving on to the next question. The question is 116. Select the true statement for AUG Okay, AUG, here we have the codon, and it is, I will also discuss the genetic salient features that are with this genetic code. Okay, so what are the salient features of genetic code? But I will not be discussing all the salient features. I have discussed them twice in previous sessions. Today I will be taking up only the particular features related to AUG. Okay, so the salient features. Okay, so the first salient feature when we discuss is the genetic code is a triplet, right? So yes, it is a triplet. We can see the 3 nitrogen bases over here, A, U and G, right? So it is present in the form of a triplet. That is very true. Secondly, we see that this particular codon, the AUG, right? It is having an exception. Generally, genetic code is degenerate, right? What do we understand by the degeneracy of genetic code? Degeneracy means one particular amino acid can be coded by more than one codon, right? If we take the example of UUU or UUC, both of them are coding for phenylalanine. That means one amino acid, okay? This is the degeneracy. But this particular codon, that is AUG, it is an exception to the degeneracy because it is non-degenerate. Okay, this particular codon is non-degenerate. Why? Because methionine is only coded by this particular codon, that is AUG. Methionine is not coded by any other codon. Okay, so degeneracy के लिए exception है हमारे पास AUG. It is a non-degenerate codon. Okay, it does not code for any other amino acid. That is true. That is applicable for all the codes. But methionine is coded by only this particular amino acid and no other. Okay, so that is why it is non-degenerate. Okay, next it performs dual function. What is the dual function? First, it codes for methionine, as I told you, and it also acts as the start codon. Wherever it will be present, the process of translation will begin. Okay? Okay, answers are there in the comment box. Answer is option 4. मैं भी देखूँ क्या है option 4, A, B and C. That is all are true for the AUG. Okay, that is true. I will go through them. But पहले इसको समझो. What are the other salient features? Okay. So it is a triplet, it is non-degenerate, it has dual function, that is it can code for methionine as well as it acts as the start codon. Okay, not the codons, it start codon. Now, AUG is basically universal, okay. So it is also following the rule of universality. Why it is universal? Because if it is coding for methionine in my body, it will code for the methionine in the body of an elephant or a bacteria or whatsoever organism you name. Okay, it will always code for methionine only, no other amino acid. Okay, so these are the salient features. Now coming to the question. First statement says it codes for an amino acid methionine. That is absolutely true. It is an initiation codon, or we can call it as a start codon. That is also absolutely true. And C is it is a universal codon, and that is also true. Okay. Otherwise, we say that genetic code is nearly universal, right? What do we understand by nearly universal? That means one particular codon, yes, it codes for say same amino acid in all the organisms, but there are certain exceptions. For example, if we take the UGA in consideration, otherwise it is a stop codon, but in case of mitochondria it codes for tryptophan. Okay, so there the universality will not stand. That is why we call it as nearly universal, okay, because there are such exceptions. But if we talk about AUG, it is absolutely universal. It does not code for anything else in any other organism. It will not act as a start or stop, it will not act as a stop codon or code for any other amino acid. Okay, clear with this? So the correct answer for the given question will be your fourth option, that is all are true for AUG. Okay, clear with this? All right. Now comes the other question, members of basidiomycetes lac. Okay, so what do we understand by when it comes to basidiomycetes members? Okay, so there are, this is again the question from biological classification. But basidiomycetes, there are, we will discuss the characteristic features of basidiomycetes. Okay, so first of all, what are the characteristics of the members of basidiomycetes? This is the class that is having the most advanced members. Okay, it is having most advanced members. The mushroom that you eat, that is the button mushroom that we speak of, right, it is belonging to this very class, right? And when it comes to the characteristics, commonly what do we call them, right? The members of basidiomycetes, commonly they it is also called as the club fungi. Okay, right, club fungi. Now the members, first of all, because it is the most advanced one, it is having the members of this particular class, they are having septate and branched mycelium. First of all, we will understand the structure only. So we have the septate and branched mycelium in the members of basidiomycetes. Secondly, we see that in the members, asexual reproduction, usually asexual reproduction takes place with the help of asexual spores. Okay, but in this case, asexual spores are absent. Okay, asexual spores are absent, and this takes place with the help of fragmentation. Right? Takes place with the help of fragmentation. Next we see asexual reproduction. After that, the sexual reproduction, if we consider. Sexual reproduction usually requires the sex organs, but in the members of basidiomycetes, sex organs are absent. Okay, so it will take place by the fusion of somatic cells. Okay, that means they are the normal cells. They are not the sex cells or the germ cells. They are the normal somatic cells of different strain. I'll show you how. Okay, pay attention to this very representation right in front of you. While we are considering, they are having the haploid phase also, they are having the diploid phase also. Okay, while we are considering the mycelium, okay, this mycelium that I spoke of, that is, the mycelium is septate and branched. Right, now this particular mycelium, one mycelium will be coming from one particular member, the other one is coming from the other one. Now what will happen? They will simply fuse with each other. Okay, suppose there is this mycelium, we know it is branched, okay, and it is coming from the other one also. This is the other parent, okay, and we have the presence of septa. Okay, now what will happen? These two mycelium will come close to each other. Okay, while they will come close to each. The whole of it has been deleted. Wow. Now what will happen exactly? while these two fuse with each other, right? They are having nucleus. They are not xenocytic, okay? Each septa is having single nucleus like this, okay? Now they are fusing with each other. Okay, now this particular part has fused. You can see otherwise there are single nucleus, right? But in this case you can see in this particular region there is the presence of dikaryon stage. It is the dikaryo phase. It lasts for a certain period, and after that these two nuclei will fuse with each other, okay? And when this structure will develop, it will lead to the formation of a basidiocarp, that is the mushroom that we see, right? These are basically nothing but the fruiting bodies, okay? Now we began from here, right? These are the two mycelium. They are coming in contact with each other, they are fusing, okay? So they are the cells that are present over here, they are the somatic cells. There are no particular sex organs that are present, right? So they are fusing with each other. It will further develop and lead to the formation of this basidiocarp, that is the mushroom. Now this basidiocarp, see, this is the haploid one, this is also haploid one. Both of them are fusing with each other to form the diploid structure. So this basidiocarp is actually a diploid structure, right? Now the basidiocarp will have the presence of basidium. The gills of mushroom that you see, आपने कभी अगर मशरूम खाया है, अगर देखा है तो आप देखते हो अम्ब्रेला लाइक स्ट्रक्चर होता है. अम्ब्रेला लाइक स्ट्रक्चर के नीचे इस तरीके से बहुत सारे स्पोक्स नजर आते हैं आपको, राइट? स्मॉल कॉम्ब लाइक स्ट्रक्चर, दैट इज द रीजन दैट दोज आर द बेसिडियम एक्चुअली, राइट? एंड दैट बेसिडियम स्ट्रक्चर विल लीड टू द फॉर्मेशन ऑफ अल्टीमेटली द बेसिडियोस्पोर्स, राइट? एंड these basidiospores are haploid spores, okay? So these are the haploid spores. From the diploid structure, haploid spores are formed. These spores again germinate to form the haploid mycelium. So this is how the alternation of the generation is taking place. Basically the haploid generation is also there, diploid generation is also there in case of this particular basidiomycetes, okay? So sex organs are absent that we saw. Asexual reproduction is taking place with the help of the fragmentation. No asexual spores are found in case of the members of basidiomycetes. Now coming back to the question, we see that the basidiomycete members lack. Okay, so here we have the sex organs. Yes, that is absolutely true. No sex organs are present in case of basidiomycetes. Plasmogamy and karyogamy, yes, it will take place. Just like I just told you, in case of the two mycelium, the somatic cells will fuse with each other. Okay, so the plasmogamy and the dikaryophase will last for a certain time, and then those also fuse. That means the fusion of nucleus, and that is the karyogamy. Fruiting bodies, yes, basidiocarps are the fruiting bodies, the mushroom I showed you in the representation, and the last one are the meiospores. Yes, these are the haploid spores that are the basidiospores, okay? So we can also call them as the basidiospores. Okay, and here this fruiting body is nothing but the basidiocarp. Okay, this is diploid, this is haploid, right? Both of them are present, and the only thing that is lacking in case of basidiomycetes are the sex organs. Okay, clear with this? Chaliye. Moving on to the next question. Select the odd one regarding arithmetic growth. So what do we understand by arithmetic growth? Okay, let us see. Then we will come back to the options. Okay, so what happens in case of arithmetic growth? Okay, let's see. If we have one cell, right? One cell will divide into two daughter cells, that we know. It has been divided into two daughter cells. Now, usually we observe the pattern that 2 will develop into 4, 4 will divide into 8, and so on. But in case of arithmetic growth, this doesn't happen. Out of two cells, one cell will differentiate. That means it will mature itself, right? It will not indulge into cell cycle or undergo cell division. Only one cell will undergo cell division. So what will happen? we observe that this cell will give rise to 2 daughter cells, right? And this is the differentiated cell. Now, next generation will see that these 2 cells will become differentiated and only single cell will again undergo division. So, what will happen? This cell will again undergo division and give rise to 2 daughter cells, and these 2 will remain differentiated. And this third one will also become differentiated, and so on. Okay, so this pattern is seen in case of arithmetic growth, and while we are plotting arithmetic growth on the graph, we observe a straight line. Right? We observe a straight line because this is a constant growth that is taking place, and for constant growth there will always be a straight line, right? And yes, linear graph, very good. Okay, now we see that the representation for this particular type of growth is by the equation LT is equal to L naught plus RT, right? Here LT is representing the length at time t, L naught is representing the length at the initial length or at the time 0, and R is representing the growth rate. Okay, so this is representing what? This is representing growth rate. Right? T is obviously time, you all already know that. Yes, okay. Now, this is the arithmetic growth. What happens in case of geometric growth? If we compare it with the geometric growth, we observe in this case that the cell that is giving rise to 2 daughter cells, these 2 daughter cells will further divide into 4 daughter cells, right? And these 4 daughter cells will further divide into 8 daughter cells, and so on, right? So we observe that initially the growth is less, right? 2 to 4 to 8 to 16, but then it grows exponentially. 16 will make directly 32, 32 will directly make 64, right? So the growth that we observe in this case will be initially slow, initially it will be slow, and then take a pace, right? And then there will come a time when the resources will become diminished, okay, or the requirements for growth. Maybe there is the nutrient medium that is present. Let's say we are observing the growth of a bacterial culture. They are growing in a petri dish. There is the presence of the growth medium, okay, nutrient medium in which the bacteria are growing. But after a certain period of time, when all the nutrient medium has been consumed, what will happen? No further growth will be observed, right? So initially there will be the lag phase, then the growth will be exponential, right? 64 to further 128, and this will be the lag, this will be the log phase, this is the lag phase, the slow growth, this is the log phase, the exponential growth, and when all the nutrient medium is consumed, it will be leading to the asymptote, right? That means the growth has now stopped. No further growth is taking place beyond this point, right? So this type of growth is the exponential growth or the geometric growth, okay? And it is represented by the equation Wt is equal to W naught ERT, okay? So these two equations, they can be asked in your exam. You should be knowing these. This is for the geometric one or the exponential one, and this is for the arithmetic one. Clear with this? Now coming back to the question, what the question was asking, select the odd one, okay, which of the following is not true with respect to arithmetic growth. So first one is only one daughter cell continues to divide while the other differentiates and mature. That is absolutely true. We just saw that, we just saw this pattern. Second, in the mathematical expression, R represents growth rate. Yes, I told you in the equation R stands for growth rate. Third one is it exhibits S-shaped curve. No, it does not exhibit S-shaped curve. It exhibits a straight line, linear graph, right? Yes, linear scale. Very good, Sagnik. And the fourth one is elongation of root at a constant rate is an example of this type of growth. Whenever we are speaking of constant growth of any plant part or any part, Okay, with that respect, that is absolutely true for arithmetic growth. Okay, so the correct answer for the given question will be your option 3, that it exhibits S-shaped curve. No, the S-shaped curve is represented by the geometric growth. Okay, clear with this? So this is all for the discussion of slot 2. This will be further carried forward by Miss Bushra, that is the slot 3. The rest of the chapters will be, the questions from the rest of the chapters will be discussed by her. So stay tuned, alright, and take care of yourselves, prepare well for your exam. All the very best.